Good morning, everybody. Welcome to the first grand rounds of the season, and also actually the first grand rounds given by the Division of Molecular Pathology and Genomics, which is the newly formed division within the department. And we've taken 4 of the grand round sessions specifically dedicated to that, although I'm sure throughout the season you'll hear many more people talking about genomics in their talks. This morning it's my pleasure to also start with one of the more youthful members of the department, Dr. Matt Meredith. And Matt is a second-year fellow in the combined Cytogenetics Molecular Genetics program, fellowship program in our department. Matt got his PhD at Rockefeller University in New York in molecular immunology under the mentorship of Dr. Michelle Nussensbach, and then did his postdoc at Harvard Medical School, continuing in molecular immunology, dealing with immune tolerance under the mentorship of Dr. Benoit and Dr. Mattis. And today, then he obviously saw the light and realized that he really needed to do some clinical work and learn cytogenetics and molecular genetics. And we're very privileged to have him as a fellow within the division. Today he'll talk to us about the genomic characterization of TALL, a tall order. Matt?
So is the volume good? So thank you, Betsy. Like she said, I'm Matt, for those of you who I have not met. And today I'm going to be discussing how we characterize TALL cases in the lab, which you'll see is a tall order. I have no disclosures. And over the course of this talk, I hope you'll be able to identify the genomic mechanisms that mediate the development of TALL, to describe TALL subtypes that depend on specific genomic abnormalities, and most importantly, recognize the prognostic and therapeutic impacts of the discovery of these recurrent abnormalities in TALL cases. So what is TALL? It's actually quite a rare subset of acute leukemias in both children and adults. In children, shown in green, we have most acute leukemias being of lymphoid origin, or ALLs, and the minority of those in orange being myeloid origin, or AMLs. This is the inverse in adults. Most are myeloid malignancies, and the minority are lymphoid. Within those lymphoid malignancies, we can subdivide 2 subtypes of ALL, whether they arise from B cells, shown in yellow, or T cells in blue. And as you can see in both children and adults, TALL represents the minority of cases in acute leukemias, such that only about 10% of childhood acute leukemias are TALLs, and even more rarely in adults, approximately 5%. So this is a rare entity. What's the big deal? Unfortunately, importantly, we have overall improvement in outcomes. This is looking at 5-year overall survival in pediatric patients. And we have seen improvement over the years. But unfortunately, the survival rates of TALL is remaining inferior to what we see in BALL. And this is likely related to the fact that it is such a rare entity that we just don't encounter it as much, and we don't understand it as well as we do, for example, BALL. And when we do encounter these entities in the clinic, how do we go about diagnosing and characterizing these diseases? As with most diagnoses, this is really a joint effort involving multiple labs, a very collaborative effort. The majority of the diagnosis is performed by heme path, looking at morphology, as well as flow cytometry looking at the immunophenotype of these cells, followed by subsequent characterization by cytogenetics looking at structural rearrangements by G-bands, more recurrent abnormalities by FISH, and more subtle cryptic copy number changes by chromosomal microarrays. Although it's currently not the standard, it's becoming an emerging effort to also implement molecular techniques to look for missense mutations, point mutations, especially in those genes that are frequently mutated in different sequencing assays, as well as identify T-cell receptor rearrangements as a measure of clonality. So by morphology, lymphoid cells tend to be smaller, with very little cytoplasm, but a large irregular nucleus, and importantly absent to indistinct nucleoli. And this profile of morphological features is consistent with a lymphoid origin and can distinguish it from myeloid, but importantly doesn't distinguish between B and T cell malignancies. For this distinction, we rely on flow cytometry looking at immunophenotype. And by flow, we look for T lineage specific markers, for example, gating on CD7 cells, and looking at the expression of other T cell specific lineage markers, including CD2, CD3, and CD1A. And in this particular case, as is often the situation in TALLs, as they are in mature T cell precursors, we often see double negative, that is, they don't express neither, they express neither CD4 nor CD8s. To really appreciate how TALL arises, it's important to recall the normal process of T cell development. So we have lymphoid progenitors that emigrate from the bone marrow to the thymus, and this is the organ in which T cell development occurs. The thymus is divided into two major parts, the cortical zone and the medullary zone. Development begins in the cortex, where early on these T cell precursors undergo TCR rearrangements that involve DNA breaks to rearrange their genome and create or generate functional TCR chains. Subsequent to that, they undergo a process of positive selection to ensure that they have a functional TCR being expressed on their surface, at which point they move down to the medullary zone and they undergo a second form of selection known as negative selection to ensure that those TCR chains are not recognizing self-antigens. And after that point, if they pass all those checkpoints, they then are able to emigrate out into the periphery in our mature T cell. So I mentioned early in T cell development we have TCR rearrangement in the cortical zone. And importantly, this process of T cell rearrangement requires DNA breaks in the genome to bring together segments of DNA that are initially separated physically in the genome, such that we now have a recombination event that brings together two segments of DNA that are now contiguous, that can then encode a functional TCR. This process requires the recognition of sequence-specific sites known as recognition site sequences, or RSS. These sites are brought together by the recombination machinery to bring together these segments, and in that process, remove the intervening sequence, circularize it, and form this T cell receptor excision circle, or TREX. And I only mention this as an interesting side note in that in newborn screening, this is the substrate that they are looking for, in that if you do not have TREX in the blood of a newborn, this is an indication that you don't have normal T cell development underway and are therefore at risk for immunodeficiencies. So I mentioned T cell rearrangements are occurring earlier in T cell development while they're still in the cortex. During the process of these TCR rearrangements, which are introducing DNA breaks. As you can imagine, if you're having DNA breaks being introduced into a cell, there's always opportunities for incorrect repair or other kind of abnormalities. So it may not be surprising that the majority of TALL cases or malignancies arise during this early stage of T cell development. The normal process of T cell development I mentioned requires these DNA breaks, because if they do not have a functional TCR, they're not going to pass, for example, positive selection. However, things go wrong all the time, and if we have, for example, a DNA break elsewhere in the genome and a DNA break at the correct place in the genome, for example, at a TCR locus, these two positions can form as substrates for DNA repair, such that we now have a translocation of this break to this other break, resulting in a transfer of material from, in this example, chromosome 10 to chromosome 14. And if this gene is an oncogene, what's critical here is that since the TCR loci are very active, highly expressive promoters, these promoters can now drive the expression of an oncogene, which can then proceed on to leukemogenesis. Worse off, in addition to targeting bona fide RSS sequences, there are also sequences throughout the genome that are very highly homologous to these RSS sites. And they can also be targeted by the machinery, such that, again, if we have a DNA break somewhere in the genome, this time within the same chromosome, those breaks can be excised and you have an intrachromosomal rearrangement. And in this situation, the intervening sequence removed a very highly expressed gene, which now places this active promoter upstream of an oncogene to drive its expression. In the course of leukemogenesis in TALLs, we actually require two major types of events. One, which are termed type A abnormalities in the literature, result in maturation arrest. These commonly involve the upregulation of a transcription factor, which normally would only transiently be expressed during T cell development, and keeping that expression going kind of arrests the cells at that stage. Importantly, there's typically a single type A event per malignancy, and these types of abnormalities are quite unique to TALL in that they often involve a translocation with one of the TCRs. And since they are unique to TALL, they can be quite beneficial, for example, in a differential diagnosis. If you're not quite sure what your malignancy, what the lineage of your malignancy is, the presence of one of these abnormalities can be quite diagnostic of a TALL. However, a type A abnormality by itself is not sufficient for leukemogenesis. In addition to that, you need one or often more type B abnormalities, which are those that increase cell survival or cell signaling. So these tend to target signaling pathways or turn off cell cycle regulators. In contrast to type A abnormalities, they typically have multiple type B events per abnormality, per malignancy. And also in contrast to type A, these tend not to be unique to TALL. They're also seen in other malignancies, especially BALLs. So I showed this example earlier. This is actually one of the more common translocations, a type A abnormality, in which the transcription factor TLX1 on chromosome 10 is translocated to the more active TCR promoter on chromosome 14, which drives the expression of TLX1 and results in maturation arrest. And what does that look like in a G-banded study? Importantly, it is visible. So if we look at chromosome 10, you can see one looks different. It has much more material. And if you look at one of the 14s, you can see one is much more truncated. So this involves translocation of material from chromosome 10, which includes this transcription factor TLX1, to chromosome 14, where we have this active TCR promoter to drive that expression. However, you might notice there's not much else going on in this karyotype. So where's the type B abnormality? For that, we need to look to other techniques. So type A abnormalities are often involving TCR rearrangements and therefore are visible in a G-banded study, whereas the type B abnormalities are often not visible and are termed cryptic because we cannot see those in a G-banded study. And to approach those, we need another technique known as chromosomal arrays. And this involves the mixture of sample DNA or patient DNA with a control DNA at a 1-to-1 ratio, each labeled in its own color, and then allowing those samples to hybridize to a chip, which contains oligos that span the whole genome. And then we compare how those two samples, based on their colors, hybridize to those positions. And if they hybridize at the same ratio, then we see that reading out the signals from those two fluorophores, they give a ratio of 1, and on a log 2 scale, that's 0. Whereas if we have loss of material in our sample, then that ratio skews to the left, indicating a loss in our sample. And conversely, if it skews to the left, to the right, that means we have more of that sequence in our sample, indicating a gain. So shown here, I have chromosome 9, and then you can see in the short arm of chromosome 9, there's this big shift to the left. And zooming in on that region, we can see this is an approximately 330 kb region. Each of these spots is a single oligo on that chip. And you can see the regions in red, and the spots in red are areas in which we have less material in our sample compared to control, indicating a loss. And this is a critical region for TALL in that it contains the cell cycle regulators CDKN2A and CDKN2B, which constitute a type B abnormality that provides a proliferative advantage. In addition to copy number changes, we also have a second type of abnormality we can detect by array, which is known as copy number neutral loss of heterozygosity. So it's copy number neutral because we still have 2 copies, but we have loss of heterozygosity because instead of those 2 copies being unique copies, we have 2 of the same copy. In a normal state, we would have 3 tracks representing alleles at which we are either homozygous for one allele, alleles in which we're homozygous for the other allele, and positions at which we are heterozygous, we have one of allele A and one of allele B. So that's the normal state. However, if we have regions in the genome in which we are only homozygous for the A allele or homozygous for the B allele, and there are no positions at which we are heterozygous, in that we don't have positions that we have one of A, one of B, this is known as loss of heterozygosity, in that previously this region had been heterozygous, but in the course of leukemogenesis, one copy was lost and the other copy was duplicated. This in and of itself is not per se a bad thing. What this implies to us is that a good functional copy of a gene could be lost, and then a mutant, non-functional copy could be duplicated. And importantly, this is again looking at the short arm of chromosome 9, similar to the copy number loss I just showed you. So this is where CDKN2A, CDKN2B maps, and this to us suggests that one or both of those genes has a non-functional copy. So we can see that arrays can provide us additional information, and we wanted to see what kind of additional information we could see in our TALL cases. And we've reviewed 20 of the most recent TALL array and G-band results. Just looking at demographically what those patients look like, consistent with the fact that most TALLs are in adolescent population. We saw the majority of our patients were quite young, with a median age of about 10, and were evenly split between females in gold and males in blue. And in comparing the number of abnormalities we could see by G bands, shown here in gold, compared to the number of abnormalities we could see by array, shown here in red, we can see that the majority of abnormalities were seen by array with a mean of about 7, whereas fewer abnormalities were detected by the G-band analysis with a mean of about 2. And importantly, you can imagine that there might be some overlap between these two studies, in that if you have a very large deletion by G-bands, you would be able to see that also in both G-bands and array. But importantly, the majority, about 90% of array findings, were cryptic and not detected in G-band studies. So we're seeing a lot more by array, not unexpectedly. So I've described to you A-type abnormalities that provide maturation arrest and B-type abnormalities that provide some proliferative advantage. Another class, or another type of finding we see in TALL cases are deletions in the TCR loci. These are normal processes in T-cell development because I mentioned in order to proceed through development, we have T-cell rearrangements that result in the excision of DNA to bring together disparate segments in the genome. The majority of these deletions in red are biallelic deletions on both copies. We had some monoallelic deletions as well as in black normal copy number, no losses at these positions. So T cells are undergoing this process normally during development, and it's a random process, so they all have different breakpoints. However, in the case of a TALL leukemia, if we have one clone that has an advantage and proliferates, then we're going to be able to see those specific breakpoints in our array, because instead of having a diversity of breakpoints, we have the same breakpoints over and over again in all of our cells. And so the fact that we can detect these breakpoints in an array of a mixed population serves to us as a marker of clonality that we're looking at a leukemic clone. Additionally, we can kind of try to harvest some additional information from copy number at these TCR loci by looking at what particular loci have biallelic or other deletions. So in the course of T cell development, we have different chains being rearranged more or less at different stages in development. The earliest being TCR gamma, followed by TCR delta. These two are required to have your gamma delta T cells. And somewhat simultaneous to that, but a little bit downstream of that, we have beginning of the rearrangement of TCR beta, followed by TCR alpha, which are important for alpha beta T cells, which are probably more familiar as they constitute the majority of T cells. So I mentioned TCR gamma is rearranged first in development. So if we look specifically at TCR gamma copy number and we focus in on the very few cells, or the very few samples, that had no copy number change at TCR gamma, which is again what we expect. Very few of these samples would have 0 copy number changes because it's the earliest chain, so it's the easiest to get to, the earliest point to get to. And among those very rare samples that had no copy number changes at TCR gamma, the other chains are also mostly unrearranged, which is again consistent with the fact that if the cell did not have the opportunity to undergo gamma rearrangement, it would not have the opportunity to undergo rearrangement of those subsequent chains. So this is consistent with arrest much earlier in maturation. Conversely, TCR beta is rearranged subsequent to those chains, later in development. And so if we look at the inverse, looking at the TCR beta locus and looking at samples in which we had either biallelic or monoallelic losses, indicating that TCR beta is beginning to be rearranged, and again look at the copy number state at those other earlier chain rearrangements, we can see a lot of rearrangements have occurred at these other chains, because in order to get to the TCR beta stage, it had to go through these earlier stages. So this is consistent with arrest later in maturation. So in addition to type A and type B abnormalities, we can also see TCR rearrangements by array, or deletions at the TCR loci that are indicative of TCR rearrangements by array. And these serve to us as markers of a leukemic clone, and can also suggest to us what stage of maturation arrest this leukemic process is at. So we can see many of these very interesting findings, but really what impact do they provide on the diagnosis and treatment of T-ALL? When we look at the recurrent abnormalities in T-ALL and look at how they were identified, be it detected by array in red or detected in a G-band or FISH study shown in gold, we can see many more. We can see much more red because we see many more abnormalities detected by array. But more importantly and more promising, I've asterisked here abnormalities that have been associated with some type of targeted therapy. Therefore, these are likely to be amenable with targeted therapies. One example of which involves a loss in the short arm of chromosome 1, a very small microdeletion, about 76 kilobases. And again, zooming into that region, we can see this loss takes out this gene, STIL1, which, as a side note, is an actively transcribed gene, as well as the promoter of this flanking gene, tau1. And the process of removing this intervening sequence places this active STIL1 promoter upstream of tau1 to drive its expression. So now we have expression of tau1. Tau1 is a transcription factor that recruits chromatin modification complexes, which then alters the chromatin state of these cells that ultimately results in maturation arrest. So it's great that we can detect these abnormalities and kind of justify what's going on in the cell, but can we really do anything about it? And importantly, there's been some very promising preliminary data from other labs showing that we can specifically target these tau1 mediated effects by inhibiting chromatin modification complexes, for example, a demethylase, shown here. So in this experiment, they transplanted primary T-ALL due to tau1 upregulation into mouse models and treated the mice without this inhibitor versus with this inhibitor. And you can see gating on those transplanted cells decreased tumor burden by treatment with this demethylase inhibitor. Importantly, this is not a nonspecific effect on any T-ALL. They also transplanted in primary T-ALL due to a different type A abnormality and did not see the same effect. So this is specifically targeting the effects of tau1 on these cells. A second interesting and also likely targetable abnormality that we've detected involves a gain in the long arm of chromosome 9. Just as a side note, you can notice that we also have a deletion in the short arm of chromosome 9 that I've mentioned before involves CDKN-A and CDKN2B. And this is a good example of the fact that many of these T-ALLs feature one or more B-type abnormalities. These are both Bs. However, the gain is much more interesting, so I'll spend a couple more seconds on that. Zooming in that region, we can see that the breakpoints of this gain map within the gene NUC214 and the gene ABL1. And whereas we often think of gains occurring intrachromosomally, this is a unique mechanism in which this segment of DNA is actually excised from chromosome 9, so deleted from chromosome 9, and circularized to form what is known as an episome. And importantly, these breakpoints come together to form a fusion transcript of NUP214 and ABL1 that encodes a chimeric protein with both of their functional domains. Another important aspect about this particular case, which is not very obvious at first, is that typically for a single copy gain, we'd expect a ratio of about 0.6. Whereas in our case, we have a very high ratio that's actually off the charts. In this case, it's expected to, based on that ratio, we should have 9 to 10 copies per cell. So instead of being a straightforward gain, this is actually what's known as amplification. We have many, many copies of this. And that is due to the fact that, as you can see from where this gain occurs in the genome, there's no centromere sequence. And because this episome lacks a centromere, it's going to be randomly segregated at cell divisions between daughter cells, such that over many generations, with more and more asymmetric divisions, you can imagine some populations of cells are going to arise that have many copies, and they're going to have a proliferative advantage. Functionally, NUP214 and ABL1 provides constitutive activation of ABL1 similar to what is probably more familiar, BCR-ABL1. So specifically, by virtue of NUP214, it's able to localize to the nuclear port complex. And by virtue of its ABL1 domains, when they are in proximity to each other, they're able to cross-activate each other and provide ABL1 signaling. However, unlike BCR-ABL1, this NUP214 fusion is actually much less active. So to compensate for that lower activity, we have this amplification via the episome, so that these fusion proteins or these chimeric proteins are able to outcompete normal endogenous NUP214 so that we can guarantee those chimeric proteins are next to each other in the nuclear pore complex and provide increased ABL1 signaling. And what's, of course, most important, multiple case reports have demonstrated that these TALLs that feature NUP214 ABL1 amplification are sensitive to tyrosine kinase inhibitors, especially to satinib. So I've shown you a couple of examples of abnormality and targeted therapy. However, it's not quite a simple A-B relationship in that we have multiple abnormalities per clone, and I mentioned you can have multiple type B abnormalities per malignancy. And many of these abnormalities play into the same pathway, so they interact with each other and can also therefore inform your therapeutic options. For example, loss of the gene PTPNN2 has been shown to actually result in tyrosine kinase inhibitor resistance in the treatment of ABL, NUP214 and BCR-ABL fusions. Similarly, Notch1 is a very common target of mutations resulting in Notch1 activation, and one of the therapies suggested to counter this activation is a class of agents known as gamma secretase inhibitors, or GSIs. However, if that malignancy also has a loss of P10, it's been shown to result in gamma secretase inhibitor resistance, despite the fact that we have these Notch1 mutations. So these abnormalities do not really exist in a vacuum. There's a lot of other things going on in these cells. In addition to seeing these really well-described recurrent abnormalities, we've also noticed a potential interesting finding among these patients. And this involves the long arm of chromosome 6, where we have well-described deletions and abnormalities in multiple malignancies, including TALL. However, these generally are large deletions, and there's no real consensus on what the critical genes are. However, we noticed many of the abnormalities in our patients shared a gene known as FIP for the plextrin homology domain interacting protein. And looking more closely at those patients in which we have some type of FIP abnormality, we notice the type B abnormalities were often P10 deletions. Out of the 4 P10 deletions we had within our 20 cases, 3 of them had some abnormality of FIP. So what is FIP? It's a good question because not many people have been looking at it. It's involved, it seems to interact with adapters that are downstream of receptor tyrosine kinases. And importantly, these go on to activate AKT and mTOR. In beta cells in the pancreas, if we knock down FIP, it's been shown to, again, knock down AKT and therefore prevent or impair cell survival. And what is probably most relevant in the context of cancer, it's been actually reported to be amplified in metastatic melanoma. And this amplification promotes a type of metabolism called aerobic glycolysis, or the Warburg effect, in which we have inefficient use of glucose instead to generate ATP to generate substrates for cell growth, including amino acids, for example. And this is a common pathway seen in many cancers. So in a metastatic melanoma, we have amplification or gains of FIP, but in our TALL cases, we're seeing losses. So does FIP decrease aerobic glycolysis, and if so, why? It seems to be a very beneficial pathway to most cancers. However, in contrast to those cancers, including metastatic melanoma, it's been shown that unrestrained aerobic glycolysis results in excessive cell stress and therefore apoptosis. So, at least in some types of TALL, it relies on this protein AMPK to basically down-modulate that aerobic glycolysis to relieve some of that stress. And in our case, we have patients with P10 loss, which would be expected to increase AKT activity and therefore also increase this pathway. But concomitant to that, we have loss of FIP, which would counter that. So perhaps FIP loss would be expected to moderate aerobic glycolysis by countering the effect of P10 loss. This is very preliminary that we need to work up more, but kind of interesting that there is some recent evidence showing that this would actually make logical sense in TALL. So in the characterization of TALL, there's no straightforward answer at this time. It really is currently a tall order. We have G-banded studies that although they detect the minority of copy number changes, they are uniquely suited to detect these type A abnormalities, which often include TCR rearrangements. And as they are unique to TALL, they provide impact for the differential diagnosis. And I didn't get to it, but they can often also be prognostic. Copy number changes that we detect by array, currently about 10% within our 20 patients, were associated with some type of targeted therapy, and this is likely to increase as we further our understanding of TALL. And I mentioned earlier there are emerging efforts to also look at point mutations in TALL. I mentioned Notch1, approximately 60 to 70% of TALLs feature some activation of Notch1, so this is a really easy target to hit. And to this end, this effort is actually currently underway here, led by Dr. Yohi, by bringing up a heme panel that includes TALL genes. So the relatively poor outcomes in TALL that we see, especially relative to BALL, really likely reflects our more limited understanding of this disease. So we need better characterization, and this better characterization will enable us to close this gap with new targetable therapies, especially when standard therapies fail. So thanks for your attention. I also want to thank my mentors, Dr. Hirsch and Dr. Dolan, Dr. Yohi for a very helpful discussion and providing the nice images of morphology and flow. The docs here at the U, as well as at the Children's Hospitals of Minnesota. and the cytogenetic staffs in the lab. So I'm happy to take questions if you have any. Yeah.
Matt, do you routinely do rearrange in all the ALL?
Not all. So it seems to be mostly the pediatric cases
Yes.
because those docs are more interested in those results. So those that we typically do not have array studies on tend to be adults.
Do you think you should?
That would be interesting because adults versus pediatric T-ALL are very different. We tend to have arrest much earlier in T-cell development in adults compared to children, which have this cortical arrest. So it would be interesting to see what additional things we don't know about those abnormalities. Yes?
Two questions for you. The first has to do with the ones where you're getting the TCR-beta rearrangements.
Yeah.
So normally RAG genes would be active. In these TALLs, are they still active and driving recombination events that could potentially cause continual...
You mean after the maturation arrest?
Yeah.
It's a good question. Likely, because, or at least some activity in some cases, because in addition to the TCR-mediated recurrent abnormalities, many of the other recurrent abnormalities that we can see emerge or evolve over time have been shown to be recurrent and depend on these cryptic RSS sites. So there might be some residual activity considering you're arrested at that stage. But I don't know for a fact.
Okay, and then the second question is, you showed how you can detect these micro deletions and how that can affect gene expression. Potentially you could get the same thing with micro inversions. Are there ways that you could detect that?
So in this case, we know that they are deleted because the copy number change is there. In order to detect more subtle inversions, there are some new techniques that are NGS-based, for example, and they look at the junctions of those novel fusions and can map where those inversions are. So yes, but that's currently not the standard. It's called, for example, mate-pair-seq. Yes?
So Matt, my question is, if you're looking at an array data in a pediatric TALL and you see, for example, something like the BCR, or excuse me, the BCR is the NUP2, can't remember.
NUP214.
The NUP-ABL1, the copy number aberration of the fusion event. So that's been described in the literature. What would you do in a clinical case if you saw a similar copy number aberration that would lead to fusions, potentially to fusions in potentially relevant genes? How do you get to a point where you can confirm if those are actually expressed and if they're actually in frame, and therefore biologically relevant? Is there a quick way, or is there something that is the normal procedure in the lab?
So you're saying if we have a unique gain that we think might result in a fusion, for example?
Yeah. Or do you just report it as a VUS?
Yes. But then also we often recommend further testing, for example, by molecular techniques, some sequencing, to see whether or not where those breakpoints occur and whether or not it's likely to form, for example, an in-frame mutation. Good question. Okay. Thank you.